A great scientific journey often starts with a single, brilliant idea. 20 years ago, a scientist named Shinya Yamanaka had such an idea. He discovered a way to turn back the clock inside our cells. He found 4 special proteins that could take an adult cell, like a skin cell, and make it young again. It was like finding a cellular fountain of youth. These proteins became known as the Yamanaka Factors. This discovery earned him a Nobel Prize. It opened a whole new field dedicated to cell rejuvenation and reversing aging. His work reframed what it means to grow old. It inspired scientists around the world. Many other labs saw the potential. We started asking big questions. If we can make a single cell young again, can we make an entire tissue young again? Could we use this to heal injuries? Could we cure diseases once thought permanent? The dream is not just treating symptoms. The dream is treating aging itself. We are exploring using these factors to restore youthful function. Work is moving from lab toward real-world applications. We are at the threshold of testing these ideas in people for the first time. Longevity science is no longer a distant dream. It is active, serious research. So, what exactly are these powerful Yamanaka factors? Think of them as a master set of instructions for a cell. They are 4 proteins named OCT4, SOX2, KLF4, c-MYC, often abbreviated as OSKM. In a developing embryo, these factors are switched on. They tell a cell to remain in a youthful, pluripotent state. This means the cell can become any type of cell in the body. As we grow and our cells specialize, these master genes are switched off. They lie dormant within our DNA for life. Shinya Yamanaka discovered we can switch them back on, even in old cells. Imagine your body's cells are like old computers. Over time, they collect errors, viruses, and junk files. The operating system gets slow and buggy. This is the epigenetic noise of aging. The Yamanaka factors act like a factory reset button. Introduced into an old cell, they begin to wipe the slate clean. When planning the very first human trial of a revolutionary new therapy, the first question is always, where do we start? For partial epigenetic reprogramming, the answer was clear. We chose the eye. The eye is an ideal place to test this technology. First, it is a self-contained system. It's partially isolated from the rest of the body. A small, protected biological island. That means an injected therapy is likely to stay there, so the risk of effects on other organs is minimized. The eye also offers a practical advantage for safety monitoring. If something went wrong, the impact would be limited. In the worst case, a patient still has their other eye. That makes the risk-benefit calculation more favorable for a first-in-human study. Doctors can easily look inside the eye to see what is happening. They can directly observe the cells and tissues. They can measure electrical signals from the optic nerve. And they can track the therapy's effects in real time. The first human trial. How the 2026 eye test will work. The process for the first human trial is designed to be as simple and safe as possible for the patients. The core of the therapy is a single, gentle injection into the back of the eye. This is a common procedure that eye doctors perform routinely. The injection delivers a harmless virus, known as an adeno-associated virus, AAV. Think of this virus as a tiny delivery truck. We have removed its own viral genes and loaded it with a special cargo, the genes for the 3 Yamanaka factors, OSK. OSK. This delivery truck is programmed to drive to the retinal ganglion cells and drop off its cargo. Once the genes are inside the eye cells, they do not turn on by themselves. They are designed with a built-in safety switch. This switch can only be flipped by a specific trigger. In this case, the trigger is a common and safe antibiotic called doxycycline. Patients in the trial will take doxycycline pills orally for a period of about 8 weeks. Our journey began with studies in mice. We created a mouse model of glaucoma, a disease that damages the optic nerve and causes vision loss, much like in humans. We injected our OSK gene therapy into the eyes of old mice with this condition. The results were remarkable. We saw a near-total reset in the cells, reversed the age-related epigenetic changes, restored youthful gene expression patterns in retinal cells. That cellular reset translated into functional improvement. The damaged optic nerves began to regenerate. Electrical signaling between the eye and brain improved. The old mice regained lost vision. We weren't just slowing the disease, we were reversing damage. Next, test in animals closer to humans. We moved to non-human primates. We used a monkey model of Nayan, a condition that causes sudden vision loss from lack of blood flow to the optic nerve. When you are working with a technology as powerful as cellular reprogramming, safety is not just a priority, it is everything. First, we used only 3 of the 4 Yamanaka factors. We excluded C-mic, reduces risk of uncontrolled cell growth. The therapy is a local injection into the eye. That keeps factors contained within a small, isolated area. Third, the drug-inducible on-off switch. We activate for 8 weeks. Then we stop by stopping the medication, long enough to reverse epigenetic aging, but too short to erase cell identity or cause tumors. If problems arise, we can immediately halt. Still, we must acknowledge unknowns. We don't know long-term effects yet. Will the rejuvenation last? These are the questions clinical trials answer. Today, March 21, 2026, we stand at a historic moment. The first human clinical trial using this technology is beginning. It marks the culmination of decades of research. We are moving from theory to practice, from mice to humans. The goal: see if we can safely reverse a key aspect of aging in people. It is a message of hope. Hope we can overcome the diseases that take our loved ones. Hope a healthier, longer life is possible for everyone. My team, many others. A couple words about the amazing work of Shinya Yamanaka and others like David Sinclair who want to extend our lifespan. In 2026, we will have the first clinical test of using Yamanaka factors in humans for the eyes. This careful, local, switch-controlled approach brings epigenetic reprogramming safely to patients. The therapy ER100 uses only 3 factors: OCT4, SOX2, and KLF4, excluding C-mic. Preclinical studies restored youthful signals and function in damaged optic nerves. The first patients enrolled in the first months of 2026, with initial results hoped for by late 2026 or early 2027. If we can safely reverse aging in one part of the body, we open the door to the future of medicine. Imagine being able to rejuvenate an aging heart, restore function to a damaged kidney, slow cognitive decline in an aging brain. The goal is not necessarily to live forever, but to extend our health span. It's about adding more life to our years, not just more years to our life. Science is a slow, methodical process. This is only the very first step on a long and challenging road. The results from this initial eye trial will be critical. The future of aging is being rewritten, and the first chapter has just begun. It promises us a not far future where we can prolong our lifespan, carefully, safely, and for the benefit of all.